So, how do you know if a person has been infected by rabies? It's really tricky. It's hard to know because we don't actually have that big an immune response to rabies, which is part of the problem. And so, if it's even suspected you might have rabies, they usually give you the vaccine and the rabies prophylaxis treatment just in case. So they can look for antibodies, they can do ELISAs to look for an immune response, but that's frequently negative even if you do have rabies. They can try to detect the virus, they can try to detect the viral genome, the RNA genome, and they can look at different tissue samples, or they can look at saliva or your central CFS. Cerebral spinal fluid. Now, again, testing for rabies is difficult because we don't have a strong immune response to it. So they frequently go ahead with the vaccine, with the shots right away. Now, as I mentioned before, this is almost always fatal after the neurologic symptoms appear, so either paralysis or changes in behavior. There have only been, like in all of history since the 1500s, there have only been 9 well-documented, verified reports of rabies survivors after neurologic symptoms started. And of those survivors, half of them had severe or permanent long-term damage. Now, in 2004, this is the Milwaukee protocol. There was a patient who is the first known to survive without getting the vaccine, and your book has a really nice kind of a focus on her that you should read about, where they used a variety of treatments in order to save her life. This is virus viral 13-1, and it details how she was exposed, and she already had symptoms or neurologic symptoms, so they couldn't give her the vaccine. It wouldn't have been effective, and so they administered several different antiviral drugs and some drugs for her symptoms and for her immune system. And now they've treated a couple other people with this Milwaukee protocol, and it's had mixed success. So that is a good thing for you guys to emphasize, for you guys to, when you're reading through, to look through that. Now, if you are in a situation like you're in animal control, you're a vet, or if you're in any situation where your chance of coming in contact with rabid animals is high, then you should definitely get the rabies vaccine. The rabies vaccine is kind of frustrating, but it only protects you for 2 years. So it's something you need to continually re-up. Our immune system really does not seem to do a good job developing immunity to this infection. Now, if you are getting this vaccine pre-exposure, you just get 3 shots, and it takes 2 weeks or so to create immunity. Now, if you're getting the vaccine after exposure, so this would be post-exposure prophylaxis, the vaccine is a lot more intense because they're not only giving you the vaccine, but they're also giving you immunoglobulins. And you need to take the whole course of immunoglobulins, and it needs to be spread out, if possible, around the site of the bite. So when you get the post-exposure prophylaxis, it's a lot of injections. You get the immunoglobulin, and that provides passive immunity until your body can actually produce an immune response to the vaccine. So you get a lot of injections of the immunoglobulin. But they can't overdo the immunoglobulin too much, because actually the too much immunoglobulin actually inhibits your immune system developing its own antibodies against the vaccine. So you get the immunoglobulin and then the vaccine is an inactivated virus. And there are a few different types of vaccines. They used to use nerve tissue vaccines, which is exactly what it sounds like. Really not any different than what Pasteur did, where they intentionally infect animals with rabies, and then they harvest their nerve tissue. And then they kind of try to make a preparation out of that and use that as a vaccine. Now, Pasteur did this because this was back in the 1800s, and they didn't have all the technology that we have now, and people were also kind of desperate because you're always going to die if you get rabies. And so his thing worked, but it wasn't the safest, as you can imagine, because it wasn't the most pure, and there was a high risk of vaccine complications. So that's still used in a few places, but now it's much more common to use vaccines that have been produced in tissue culture. So either using egg embryos to grow the virus, or in tissue culture, and then you inactivate that virus. And this can be produced in a much more controlled way, and it can reach a higher level of purity. And so these vaccines, like in the States, we use inactivated virus from that's been grown in tissue culture, and this is much safer. Now, that being said, if this was 1905 and I had just been bitten by a rabid dog and I had a 20% chance of an adverse reaction to a nerve tissue vaccine in 1905, but I'd have a 100% chance of dying without it, yes, I would take that chance and take the vaccine. But nowadays, we have the cell culture vaccines that are better and safer. Now, after you're bitten by a rabid or suspected rabid animal, they clean the wound. They used to cauterize it, hopefully killing the virus. Now we wash it, and we can use like iodine solution or so forth to hopefully kill the virus. But then you're given the post-exposure prophylaxis. So it's a lot of injections of immunoglobulins, and then they follow up with injections of the inactivated virus, the vaccine. And this treatment can be expensive, and this can be cost prohibitive in some places of the world. And so unfortunately, in those situations, the person just dies. They do what they can to make them comfortable. The Milwaukee protocol has had mixed results, mixed success, but also a lot of those medications can be expensive. So if you are in an impoverished area of the world, all they can do is make you comfortable, and then you die. So looking at the virus on a more molecular level, right? This is in the genus of lyssavirus. There have been some other lyssaviruses that have been found circulating, some of which have been pathogenic, but they are much less common. Rabies virus has a really unique bullet shape, so kind of rounded on one end. They really do look like little bullets. And these viruses are covered with glycoprotein spikes, which is kind of illustrated here. They look, it's not how I would draw a glycoprotein, but they look kind of decorative and pretty in this cartoon. And these glycoprotein spikes actually have homology to snake venom, and they bind some neurotransmitter receptors on the surface of your nerve cells, which can lead to some of those changes in behavior. Now, the glycoprotein is embedded in the envelope. The envelope, underneath the envelope, we've got the matrix protein, which covers the genome, the RNA genome, and the RNA genome is coded in nucleoprotein. And then it's an RNA virus, so it's got to bring in its own RNA-dependent RNA polymerase, and that's the L protein. And a lot of viruses, you might notice as we're going through the semester, the polymerase is referred to as an L. And that's often because a lot of genomes are segmented, and you've got a short fragment and a long fragment, and the long fragment usually is the polymerase. Now, the rabies virus is not segmented, it's continuous, but we still refer to the polymerase as the L gene. It's one of the longer genes. So it is a single-stranded negative RNA virus, and it has an interesting architecture in that it doesn't synthesize one pre-protein that then has to get cleaved, or that multiple transcripts come off of. It has these intergenic regions of a few nucleotides that serve as stop points. So the polymerase will do the first gene, and then it'll stop, and then it'll do the next gene, and then it'll stop and do the next gene, and it'll stop. It does have one really long intergenic region that's theoretically long enough to be a gene, but as far as we know, it's not a gene. So it's possible this is just a really long spacer. It's possible this used to be a functional gene, but it picked up some mutations and it's not anymore. It's possible it does something that we don't know what it does. Our functional genes, we've got N, our nucleoprotein. This coats the RNA genome and protects it from being recognized by the innate immune system. Our phosphatoprotein, this is actually a really busy protein. It does a lot of things in the rabies replication cycle. So it helps stabilize the RNA polymerase during replication and transcription. It binds to the soluble nucleoprotein, and it prevents it from encapsulating RNA before its time. So it makes sure that only genomic RNA gets encapsulated and not messenger RNA. Also, this phosphatoprotein, it can antagonize the innate immune system, so it prevents the host interferon alpha and interferon beta signaling. And also, it's possible that the phosphatoprotein helps the virus traffic through the cell towards the cell body. So this protein does a lot. It's a very multifunctional protein. Then we've got our matrix protein. The matrix protein is responsible for budding the new viruses from the infected cell. It's also important for helping to maintain the bullet shape of the virus. It completely covers the nucleocapsid and the genome, so it maintains the shape of the virus and it helps with budding. And then we've got our glycoprotein that's going to be responsible for binding to the surface of the cell, and then fusion, so entry. And then we've got our L protein, and L protein is the polymerase, and again, this is a multifunctional protein, so it's going to be responsible for replicating the genome, it's going to be responsible for transcription, and it's going to be responsible for processing those messenger RNAs. It's going to add the poly A tail, and it's going to add the 5' cap. So we've got lots of multifunctional proteins here, characteristic of a virus. You've only got 5 genes, so you've got to make the most of what you got. Looking at the viral replication cycle. So, of note that I mentioned before, the glycoprotein is homologous to snake neurotoxins. We think that this is responsible for some of the changes in behavior that are witnessed in patients. It binds to nicotinic acetylcholine receptors. The entry is, so it comes in through endocytosis. It's in the endosome. The low pH of the endosome triggers fusion. And then the M protein dissociates during uncoating, and you've got the core of the virus. It needs to travel up the axon to the cell body. So this is actually going to hijack retrograde transport, because it can't do any replication here. It can't replicate itself. It can't make proteins here. It's got to travel up to the cell body. Now, then we've got to undergo transcription. So this is dependent upon L, our RNA-dependent RNA polymerase. And it's a negative single-stranded RNA virus. So first you've got to make the sense strand. That sense strands can be used as messenger RNA. And then in order to make more genomes, you've got to run copies off of that sense strand. And in order for those messenger RNAs to get translated by the host ribosome, they need a cap and a tail, which the L protein or the polymerase also adds. Protein synthesis. All right, so the proteins that don't need further processing are translated by free ribosomes just in the cytoplasm. The glycoprotein needs to be glycosylated. So it gets translated on a ribosome that's attached to the ER, the rough ER. Then it goes through the Golgi, where it's glycosylated, and then it gets sent to the plasma membrane. And then the matrix protein is going to kind of line the membrane. Everything's going to congregate together there, and the matrix protein will help it bud. Now, timing of this is sort of regulated. So genome replication. So when the virus is still in the process of in the middle of its replication cycle, you make the positive strand, and you're going to make lots of messenger RNAs, and some of those messenger RNAs are going to become nucleoprotein. When you have enough nucleoprotein, that triggers the virus to switch to genome replication. So instead of making more messenger RNAs, then you start transcribing the genomic RNAs, the negative sense full-length genome. Then everything, the genome, the glycoprotein that's gone through the ER and the Golgi, the matrix protein, all of that comes together under the plasma membrane and it buds off. So the virus steals its envelope from the plasma membrane. And that's the replication cycle in nerve cells. We know the virus replicates in muscle cells for a while before it goes to the central nervous system. We know that it antagonizes the innate immune system. We know that it can downregulate the interferon response. We don't entirely understand why the virus doesn't generate a lot of antibodies. It's not very antigenic. And during the course of infection, if you did an ELISA, you might not find any IgG or IgM in the patient at all. That might get to the neurologic phase, and they might have some IgM in their serum, but not in the cerebral spinal fluid. And the virus also does another odd thing. People have really gone back and forth about this. It's possible if they have a cytotoxic T cell response, that might destroy some of the infected cells, but we don't think the virus actually kills the cells. The virus does not induce apoptosis. They've done a lot of studies where they've, after death, they slice open the brain and they look for signs of pathology. And even when you can find the virus, like you can stain for it, section it, stain for it, you can find the virus really widespread in all these nerves. There's actual very little damage to the brain itself, which is really odd. You would expect like really widespread cell death, but we don't actually see that. So rabies virus is and it's an interesting virus. It's not super duper common in the States. As long as you avoid rabid animals, you should be fine. no reason to worry about it. But if you are bitten by an animal, or a rabid animal or a suspected rabid animal, it's really, really important to make sure that you get the the vaccine, because without the vaccine, you will not develop an antibody response to this virus in time. Because for whatever reason, we don't seem to do that on our own very well at all. When the virus is replicating inside your nerve cells, it really kind of lays low, and it might, shut down a lot of the processes within your cell. But it's not causing widespread damage to your brain. It's not causing a huge immune response. It's not causing massive inflammation. It really doesn't elicit much of an immune response, which we think is one of the sneaky kind of difficult things about it. It does stimulate parts of your brain that lead to that increased salivation, to more biting, to being more aggressive. You're often not afraid of the dark anymore. You're not afraid of other animals anymore. And so the way it spreads, right, is through that saliva and through that biting. So through the course of infection, you are sort of encouraged to go out there and bite other animals. Almost kind of like a zombie virus. I think a lot of zombie movies were kind of based on a very fictionalized, dramatized version of a rabies virus. Anyway, with this, I'll end. If you guys have questions, let me know. Send me an email or stop by in the chat. Hope you guys are doing well.